Good morning, student. Today we will discuss about inhibition of enzyme action. You have already completed the basic of enzyme, its kinetics, its properties, etc. So today we will discuss about the inhibition. Enzyme is one of such topic that is very difficult to teach without interacting with the students. So it's really difficult for us to, but it is the need of the hour, so we have to. Hope you will all be with us. So let's start. In this topic, what we will study? We will study the enzyme inhibition, its importance, its various types and uses. Okay. Now, what are enzyme inhibitors? Enzyme inhibitors are molecules that interfere with the catalysis process either by slowing or by halting the enzymatic reaction. Okay, that means in our body system there are so many reactions that is catalyzed by one or other enzyme. So if we inhibit the enzyme action, that means we can slow down the rate of catalysis, or we can completely halt the enzymatic reaction with the use of enzyme inhibitors. Okay, so what is the importance? Why we should study enzyme inhibition? Most of the pharmaceutical agents known till now are enzyme inhibitors, because you all know there are so many processes going in our body, and one or more are involved in some pathogenesis of some kind of diseases. So to prevent the disease, that particular disease, we have to identify the key enzyme and we have to halt or slowing the reaction. So enzyme inhibitors is one of the most popular and known pharmaceutical agent. Okay, other than this therapeutic use, there are diagnostic use too, because to delineate some metabolic pathway, suppose glycolysis, which step will come after which step. To delineate, this is the first step, this is the second step. To delineate the metabolic process, the enzyme inhibitors are very much useful, because if we inhibit particular enzyme, the product before that step will accumulate, and if you can assay the product, then we can develop, or we can chalk out the gradual process, step by step process of the metabolic pathway. Okay, so the enzymes inhibitors are important both from therapeutic point of view and from diagnostic point of view. Okay, basic core biochemical process can be delineated by the use of enzyme inhibitors. Okay, so what are the types? Two broad classes, reversible and irreversible. Under reversible, there are competitive, uncompetitive and mixed. Please remember these classification, because in your textbook, that is you people commonly going through the Vasudevan, isn't it? So in this type of book, there are some wrong perception, that is reversible. Under reversible, there are competitive, uncompetitive and non-competitive inhibition. But please remember this classification. This is the classification of any standard textbook, and this is the scientific one. So please give attention to this type of classification, because it is very common question in your both in theory and practical and viva. So you should classify the enzyme inhibitors like this. That means inhibitors are classified reversible and irreversible. Under reversible, there are competitive inhibition, uncompetitive inhibition and mixed type of inhibition, and non-competitive inhibition is a special type of mixed inhibition. Okay, non-competitive and irreversible is not the same, which is generally mislead this your book like Vasudevan mislead to the idea that non-competitive inhibition means the irreversible inhibition. Not that. Okay, non-competitive inhibition is a special type of mixed inhibition. We will discuss later on. So the broad classification of enzyme inhibitors are competitive, uncompetitive and mixed inhibition, which comes under reversible, and another is irreversible. Okay. So let's see one after another. So the first is competitive inhibition. You all have going through the competitive examination, isn't it? Competition means more than one person compete for a single place, isn't it? So here also competition between what? Competition between normal substrate of enzyme and inhibitor. Okay, how inhibitor molecule able to compete with the normal substrate of the enzyme? Because the inhibitors are structural analog, structurally very similar to, structural analog means structurally very similar to the substrate. So inhibitor compete with the substrate for the active side of the substrate binding side of the enzyme. Okay, and preferentially binds with the enzyme, because if the substrate can bind with the enzyme, inhibitor can also bind with the enzyme. But what will be the result? If substrate bind with the enzyme, then enzyme plus substrate, enzyme substrate complex will form, and that leads to product formation of the product. But if inhibitor succeed to overcome the substrate and preferentially bind to the enzyme, then enzyme plus inhibitor, enzyme inhibitor complex will form, and that leads to no production of any viable product. Okay, so reaction will be halted. That is the basic of competitive inhibition. Okay, let's me activate my marker. Okay, so in competitive inhibition, inhibitor will be structural analog. One criteria is structural analog of the substrate. Inhibitor molecules compete with the normal substrate molecule for attaching with the active site of the enzyme. So E plus ES complex can be produced, which leads to enzyme plus product, and E plus EI complex can be produced, which leads to no product development. Okay, so number of enzyme molecules available for ES formation is reduced. Why? Because substrate and inhibitor both compete for the same place. Okay, so if inhibitor is present, some of the inhibitor molecules will surely binds with the enzyme. So ES formation must be reduced. If not completely abolished, but must be reduced, because inhibitors are there. So inhibitor compete with the substrate. Some of the inhibitor succeed and they bind with the enzyme to form the EI. So the enzyme molecule available for binding with the substrate will decrease. Competitive inhibition is usually reversible. It comes under reversible. Why it is called reversible? Because if we add excess amount of the substrate, that means substrate is much more higher in concentration than the inhibitor molecule, then almost all enzyme will occupied by the substrate, then the inhibition by the inhibitor can be overcome. Okay, but okay, let's see this picture. It's clearly demonstrated. This is the enzyme molecule and this is the substrate binding site. Substrate and inhibitor are structurally similar. See here the binding with the enzyme, that is the enzyme active substrate binding site, active catalytic site, these both are looking similar. Okay, so they are structural analog, and enzyme can bind with the substrate to form ES or inhibitor to form EI. Okay, now here is one example. Here normally succinate is converted to fumarate. Succinate formula is CH2COH, CH2COH. The competitive inhibitor of succinate to fumarate formation is malonate. Because see here malonate is structural analog to succinate. Here CH2COH, here also CH2COH. Okay, so substrate enzyme substrate binding site can bind succinate or can bind malonate. So malonate is one of the competitive inhibitor of succinate to force succinate leads to formation of the fumarate, but if malonate binds with the enzyme, no formation of fumarate. Okay. Now, what will be the effect on Km and Vmax? That is very important. What is the effect of competitive inhibition on Km and Vmax? You all know what is Km. Km is the substrate concentration where the velocity, reaction velocity will be the half of its maximum velocity. And what does Km denotes? Km denotes the affinity of the enzyme towards the substrate. So, you all know that if Km is higher, that means affinity is low. That means more substrate is required to saturate the enzyme, and Km is low means less amount of substrate is required to saturate the enzyme. That means the affinity is high. Okay, so reverse. Okay, now here what happened? Due to the presence of inhibitor, the substrate affinity of this enzyme towards substrate will decrease because it's preferentially it can either binds with the substrate or binds with the inhibitor, isn't it? So the apparent Km will be increased. That means Km increased means affinity decreased. Apparent Km will be increased. Why it is called apparent? Because if we increase the substrate concentration much more higher than the inhibitor, then enzyme will binds with the substrate preferentially. Okay, so apparently in presence of inhibitor, some of the enzyme molecule obviously occupied by the inhibitors, and much more substrate is needed to occupy the same amount of enzyme, isn't it? That means apparently Km is increased in presence of competitive inhibitors. In other words, the affinity of the enzyme towards substrate is apparently decreased in the presence of inhibitors. But what will be the effects on Vmax? We have already known that if we increase the substrate concentration, enzyme will saturate with the substrate and the reaction can be occurred in its maximum velocity. So Vmax remain unchanged. Vmax is not changed. Okay, Vmax is not changed because substrate, by increasing the substrate concentration, we can overcome the inhibition. So Km is apparently increased because much more substrate is required to occupy or saturate the same amount of enzyme. That means affinity of the enzyme towards substrate is apparently decreased. So Km is apparently increased, but Vmax remain unchanged. Vmax is not changed. Please remember this. For every inhibition, this is very important. Okay, and try to practice drawing both these curve. That means, that is, you all know what is this curve? Lineweaver-Burk plot, and this is Michaelis-Menten plot, isn't it? Please practice drawing these two curves, Lineweaver-Burk and Michaelis-Menten plot, with each inhibitors. Okay, each type of inhibition, competitive inhibition, uncompetitive inhibition, non-competitive inhibition, everywhere. Okay, it is very important. So here is the depiction of the curve for competitive inhibition. This blue line denotes the enzyme reaction without inhibitor. If inhibitor is added, then what happened? Here 1 by Vmax, Vmax remain unchanged. So here also Vmax point is same. This is the Vmax point, but Km increased. Km increased means minus 1 by Km decreased. Okay, so this is the Lineweaver-Burk plot, and here depiction of the Michaelis-Menten plot. Okay, if we can stop this, then what will happen? The bacteria cannot synthesize the folic acid, so They cannot synthesize their genetic material and they will die. And sulfonamides just look like para-aminobenzoic acid. It is structural analog. See, it's the structural analog, structurally very similar to para-aminobenzoic acid, which is one of the substrate for synthesis of the folic acid. So sulfonamides group of antibiotics, they competitively inhibits the bacterial folic acid synthesis. Okay. So bacteria cannot synthesize this folic acid, their genetic material, and cell wall is impermeable, so it cannot absorb folic acid from outside, and it is absolutely harmless to human being, because in human being there is no machinery for synthesis of the folic acid in vivo. That's why it's preferentially, selectively, they destroy the bacteria and acts as antibacterial agent. Okay. Now, methotrexate. Methotrexate is very well known anti-cancer agent. Anti-cancer agent. What will happen in the cancer? Cancer means uncontrolled proliferation of one's particular cell, one's own cell, uncontrolled proliferation. That means there is so many first cell division, uncontrolled cell division. That means DNA synthesis is very essential. So here also we need folic acid. Here what happened, methotrexate is chemically, it is 4-amino-N10 methyl folic acid. 4-amino-N10 methyl folic acid. So it is structural analog of folic acid. In DNA synthesis or folic acid DNA synthesis, there are some steps. Pyrimidine synthesis, there are some steps where folic acid is first converted to dihydrofolate, then to tetrahydrofolate by dihydrofolate reductase and folate reductase and dihydrofolate reductase enzyme. Folic acid to dihydrofolate by folate reductase and dihydrofolate to tetrahydrofolate by dihydrofolate reductase enzyme. So if methotrexate acts as analog to folic acid, these steps will not occur. Folic acid cannot be converted to dihydrofolate reductase, which is very necessary. That's why it halts the DNA synthesis and thus stop the cell division. Therefore, methotrexate is very useful as anti-cancer drug. Next one we should mention is dicumarol. Dicumarol acts as anticoagulant. Vitamin K is very much necessary for clotting mechanism. Clotting factors cannot act properly without presence of vitamin K. So if one agent which is structurally similar to vitamin K can provide from outside, they competitively inhibit the process. That means vitamin K is essential for the clotting, so its inhibitor, it will act as anticoagulant. So dicumarol structurally similar to vitamin K, and thus it's inhibit the process of clotting and acts as anticoagulant. Okay. And there are so many isonicotinic acid hydrazide INH is very well known antitubercular drug. It also acts with the main mechanism of competitive inhibition. There are so many examples. By utilizing this competitive inhibition, principle of competitive inhibition, we successfully treat patient who ingest methanol. Methanol is a solvent found in the gasoline antifreeze. So if one ingests methanol, then methanol is one type of alcohol. So methanol is first converted or oxidized to aldehyde with alcohol dehydrogenase, then from aldehyde to acid. So methanol to formaldehyde to formic acid. You know better than me because you have just passed your chemistry exam in your 12th class. So methanol is with the help of alcohol dehydrogenase converted to formaldehyde, then with the aldehyde dehydrogenase converted to formic acid. This formaldehyde is very toxic because our eyes are particularly very sensitive to formaldehyde poisoning. So blindness can occur if one ingests the methanol which found in gasoline antifreeze. Ethanol also is a natural substrate of alcohol dehydrogenase. Okay, methanol is also alcohol and ethanol is also an alcohol, so both are substrate for alcohol dehydrogenase. But ethanol is converted to acetaldehyde. Instead of formaldehyde, ethanol is converted to acetaldehyde, which is not toxic to our body. Here, if we gradually infuse the ethanol in a metered dose or clinically calculated dose to maintain the blood level of ethanol in a stable condition, then the alcohol dehydrogenase preferentially acted over the takes the ethanol as substrate and converted it to acetaldehyde rather than taking the methanol as substrate. So the formaldehyde already formed receive enough time to get cleared via kidney. Okay, so toxicity reduced. Methanol toxicity, effect of methanol toxicity due to formation of formaldehyde reduced, because by using the ethanol, which is another substrate, natural substrate for the enzyme alcohol dehydrogenase, the alcohol dehydrogenase preferentially take up the ethanol from the media and convert it to acetaldehyde, which is harmless or non-toxic, and the formaldehyde already formed due to ingestion of the methanol get enough time to clear out the body, and excess formaldehyde formation also preferentially stop. So here ethanol almost acts like competitive inhibitor, isn't it? Though it's natural substrate, but here ethanol acts as competitive inhibitor, compete with the methanol for the enzyme alcohol dehydrogenase. Okay, let's see the other types of inhibition. The other type of inhibition, irreversible inhibition, that is, we have already told, that is uncompetitive and mixed type of inhibition. Though they have been explained with the example of single substrate, but actually it is observed only with the enzymes having two or more substrate. For better understanding the mixed and uncompetitive type of inhibition, also explained with the help of one substrate, but naturally in our physiological condition it occurs only in case where the enzyme having two or more substrate. Okay, so uncompetitive inhibitors, what happened in uncompetitive inhibition? Enzyme can bind with substrate to form the enzyme substrate. After enzyme bind with the substrate, then only inhibitor can bind with the ES complex. Unless the ES complex is formed, inhibitor cannot bind. Why? Because binding of the substrate with the enzyme causing conformation change in the enzyme to open up the binding site for the inhibitor. So substrate first bind with the enzyme to form ES and leads to conformational change, so that the place for binding of the inhibitor open up, and then inhibitor can bind to form the ESI complex. Okay, so what happened here? Definitely there are two separate sites for binding of the substrate and inhibitor. Not that like competitive inhibition, the single site substrate and inhibitor compete with the same site. Not that. Substrate has its own binding site and inhibitor has its own binding site, but that these inhibitor binding site open up only after formation of the ES. This is the peculiarity or typical nature of uncompetitive inhibition. Okay, so here EI complex cannot be formed, either ES or ESI. Okay, in competitive inhibition ES or EI, but here EI, enzyme inhibitor complex, cannot be formed. Enzyme substrate inhibitor complex can form. Okay, so if there is ES can lead to E plus P or enzyme plus product, or if ES binds with the inhibitor, then ESI, which leads to no product formation. Okay, so Let's see. Binds at a safe site distinct from the substrate active site, binds only to S complex, lower the measured Vmax. Why lowering the measured Vmax? Because here increasing the substrate concentration cannot overcome the stop the binding of the inhibitor, because they are a separate site, and ES formation leads to or potentiate the binding of the inhibitor. So they are a separate site for binding of the substrate and inhibitor. So increasing the concentration of the substrate cannot cannot overrule the action of inhibitor. So Vmax in a reaction can occur, but Vmax cannot be reached. So Vmax lowers the measured Vmax. Okay. Apparent Km also decreased. Km decreased means, what is the meaning of decrease of the Km? Affinity increase. So how inhibitor increase the affinity? Isn't it something fishy? Sound like some, how it could be possible one inhibitor can increase the affinity? Here Km also decreased because the concentration of substrate required to reach 1/2 Vmax decreased. Why? Why? Because here ES is channelized to formation of the ESI, and this ESI cannot produce any product. So there is no product inhibition. Once ES is formed, then ES can bind with the inhibitor, and enzyme substrate inhibitor complex will form. So no product development, but substrate is utilized. So enzyme think, yes, substrate is right, but no product is formed. So there is no product inhibition. Rather, you have to produce the product. So that is why affinity towards the substrate apparently increase. That means Km is decreased. So apparent Km also decreased because the concentration of substrate required to reach 1/2 Vmax decrease. Okay. Example of uncompetitive inhibition. Inhibition of the placental alkaline phosphatase. Alkaline phosphatase is an enzyme which has various types of isoenzyme and isoforms. So placental alkaline phosphatase, which is also called Regan isoenzyme, it is inhibited by phenylalanine. Phenylalanine. This is one example of uncompetitive inhibition. So in uncompetitive inhibition, what happened? So in uncompetitive inhibition, what happened? That is Vmax is decreased and Km is also decreased. That means apparently affinity is increased. Why? Because first substrate binds with the enzyme and substrate is removed from the media without formation of the product, because ES complex binds with the inhibitor and no product will form. So apparently it appears that substrate is preferentially occupied by the enzyme. So apparently affinity towards the substrate is increased. That means Km is decreased. Okay. Now in mixed inhibition, what happens? What is the difference between uncompetitive and mixed inhibition? In both cases, there are separate site for binding for the substrate and inhibitor. The substrate binding sites and inhibitor binding sites are separate. Okay. The inhibitors are not structural analog of the substrate. In both cases, inhibitor are not structural analog of the substrate. But what is the difference? Here, the binding site for substrate and inhibitors are separate. Not like the uncompetitive inhibition, where after binding of the substrate only, the site for binding of the inhibitors is open up, functional. Not like that. From the very beginning, the enzyme can bind with the substrate or enzyme can bind with the inhibitor. Anyone. Okay, enzyme can preferentially bind with the inhibitor to form enzyme inhibitor complex, or it can preferentially bind with the substrate to form the enzyme substrate complex. And this enzyme inhibitor complex can again combine with the substrate, because substrate binding site is difficult to form enzyme substrate inhibitor complex, or the first produced enzyme substrate complex can bind with the inhibitor later to form enzyme substrate inhibitor complex. Okay, so here this is the mixed type of inhibition. Why it is mixed? Because enzyme in either way it can produce ESI, not that E plus S then I only. It can produce E plus S then I, or E plus I then EI plus S to form ESI. Okay, so it is called the mixed type of inhibition. And non-competitive inhibition is a special type of mixed inhibition where this value of K1 and K1 prime is equal. Okay, where the value of K1 and K1 prime is equal, that is called non-competitive inhibition. You need not to remember this K1 and K1 prime things. It is not in your syllabus. You just remember, in non-competitive inhibition there are separate site for substrate binding and inhibitor binding. They are substrate and inhibitor are not structural analog. Enzyme can preferentially bind with the substrate first or the inhibitor first. In both cases, they binds with the other to form the enzyme substrate inhibitor complex. So here the curve, Lineweaver-Burk plot, and the Michaelis-Menten curve for this reaction, this type of inhibition. You have to practice this also. So what will be the result of Vmax and Km in case of mixed inhibition or non-competitive inhibition? Here you can say that Vmax will decrease. Obviously in uncompetitive and mixed or non-competitive inhibition, Vmax obviously decrease, because increasing the substrate concentration will not not overcome the reaction mechanism, isn't it? Because substrate binding site and inhibitor binding site is separate. So inhibitor and substrate, there is no competition between inhibitors and substrate. So increasing the concentration of one, we cannot overcome the others, the effect of other. So here obviously Vmax will decrease. Okay, reaction velocity, because some of the enzyme molecule must be occupied by the inhibitor, which doesn't lead to product formation. So available enzyme molecule for the substrate was obviously decreased, because they are separate binding site. So some of the enzyme molecule occupied by the inhibitor, which cannot be overcome by the increasing the concentration of the substrate. So the reaction velocity obviously decreased. But what will be the effect on Km? Obviously Km remain unchanged here. In non-competitive inhibition, Km remain unchanged. Why? Because there is separate site for binding of the substrate and inhibitor, isn't it? And not like the uncompetitive inhibitors also. In uncompetitive inhibition, first you have to produce the enzyme substrate complex. So the substrate is removed from the media. Apparently it seems the substrate is removed from the media. So binding is increased without production of the formation of the product. Enzyme substrate complex binds with the inhibitor to form ESI. So their affinity is apparently increased. But here any of the reaction, that is enzyme plus substrate or enzyme plus inhibitor, any of the reaction can occur first or simultaneously. Okay, so no effect of affinity, no effects on affinity of the enzyme towards the substrate. Simply that it doesn't affects on the enzyme towards its affinity towards the substrate. Okay, so the Km remain unchanged. Km remain unchanged. Now here also you see, here is the Km. Km in the same point, it would say with inhibitor, it is without inhibitor, blue line without inhibitor. So here Km remain unchanged, but Vmax decrease. Vmax decrease, that means 1 by Vmax will increase. Vmax decrease, that means 1 by Vmax increased. So with inhibitor 1 by Vmax is increased. In Michaelis-Menten curves also, here highest point is here in the, this is blue line is without inhibitor and red line is with inhibitor. Okay, so for blue line without inhibitor, here is the Vmax. So half Vmax is here. So if you draw a vertical line towards the x-axis, this is the Km. Okay, and for with inhibitor, that is this red line is with inhibitor, this is the Vmax. So this is the half Vmax. Here also Km remains same. Km remain unchanged. The affinity of the enzyme towards substrate will not be altered. Okay, but what happened? The Vmax is decreased. Vmax is decreased. Okay. Okay, it will be very helpful. Actually, it is very really frustrating to teach you in this online mode this type of inhibition. Actually, if you draw it in the board and ask you and tell you, ask me the question, that will be very easy for us, and that we as teacher become very happy. It's really not up to the mark to teach you these topics in online platform. Anyway, whatever. So hope in your revision class, if you have any doubt, we will clear it out in offline mode. So mixed inhibition we have already discussed. So what is non-competitive inhibition? In special case, rarely encountered in experiments where Km value of both the reaction of formation of E plus S and E plus EI are same. This classically has been defined as the non-competitive inhibition. You need not to say this, you just say the basic thing, that is there are two separate sites for binding of the substrate and the inhibitor, and any of them can preferentially binds with the enzyme first and leading to formation of the, ultimately leading to formation of the ESI complex, and Km remain unchanged, Vmax decreased. Okay, now you can easily can conclude that increasing in the substrate concentration gradually does not relieve the inhibition. Need not to explain it anymore, because you I think it's clear. So some examples: cyanide inhibits cytochrome oxidase, which is very important enzyme of the one of the complex in electron transport chain. Cyanide poisoning can be life threatening too. Now second one is fluoride. Second example is fluoride. This is very interesting. Okay, and this is for a practical experience. Okay, you will every time you will draw blood in your internship. Every time you see or prescribe the blood for blood glucose, fasting blood glucose, postprandial blood glucose, you have to collect the blood sample in the fluoride vial. Okay, so why fluoride is used? Fluoride actually removes two important ions, magnesium and manganese ion, and inhibit the enzyme enolase. Enolase is one of the very important enzyme in glycolysis pathway. Glycolysis you all have studied in your 12th class. In glycolysis means metabolism or catabolism of glucose, isn't it? So if you wish to determine the value of blood glucose and the glycolysis occur, then what will happen? You will falsely get the lower value, isn't it? Because glucose is utilized, catabolized. So when sample is drawn and when test is done, during this time period, between this time frame, the blood glucose will be utilized or degraded or catabolized, so you will get false lower value of blood glucose. Okay, and blood glucose is in determination of the exact value of blood glucose is very important. You all know that we can say the most common test one physician prescribes for fasting blood glucose, postprandial blood glucose, isn't it? You have all very familiar with the term. So fluoride, by inhibiting one of the important enzyme in the glycolysis system and removing the magnesium and manganese ions, which are very important for functioning of the some other enzyme too in the glycolysis pathway, it inhibit the glycolysis process. So glycolysis process is inhibited, that means glucose is not utilized or catabolized, and we will get the exact value of blood glucose. That is the reason blood sample for estimation of blood glucose is collected in the fluoride vial. Fluoride is added in the vial, and in that vial only we should collect the blood glucose. Okay, serum for estimation of the blood glucose. Now another example is iodoacetate, which inhibit enzyme having sulfidyl group in their active center. Various enzymes have sulfidyl group in their active center, and iodoacetate inhibit those type of enzyme. British antileucide or dimercaprol. This one is also very interesting. British antileucide, BAL or dimercaprol. This is used as an antidote for heavy metal poisoning. You are all familiar with the name heavy metal poisoning. Some heavy metal lead, mercury, they are toxic to our body. This color paint has have plenty of lead in it. Color pencils, color cheap color paints, toys, even the color used in the toys, plenty of leads are there. And there are so many heavy metals in our environment, daily day-to-day reagents, isn't it? And if these heavy metal causing poisoning, the British antileucide or dimercaprol is the antidote. That means it reverses the poisoning effect. How? Heavy metal acts as enzyme poison by reacting with the sulfidyl group. Various enzymes have sulfidyl group, we have discussed in the iodoacetate, and heavy metal react with this sulfidyl group and inhibit those normal functioning of the enzyme. Thus it is very much slowly heavy metal cause poisoning, isn't it? So sulfidyl group is it reacts with the sulfidyl group of the enzymes and inhibit or halting or the action of that enzyme. British antileucide, it also have got plenty of SH group in within it. Okay, so heavy, when BAL is administered, then heavy metal think these sulfidyl groups are plenty, so we can bind with this sulfidyl group. So instead of binding with the normal enzyme in our body or physiological system, it preferentially binds with the sulfidyl groups present in the British antileucide, okay, dimercaprol. So the poisonous effect is reduced, isn't it? By this way it acts as antidote for heavy metal poisoning, and all these are example of mixed type of inhibition. Okay, or you can say non-competitive. If in your exam give the question is like that, give the example of non-competitive inhibition, you can also give these examples: cyanide, fluoride, iodoacetate, dimercaprol. Okay, now after completion of the reversible inhibition, let's see what is irreversible inhibition. You can get an idea from the name. Irreversible means it cannot be reversed back. The enzyme function when inhibited, it cannot be reversed back until the new enzyme is formed. So the molecule of enzyme is inhibited by the process of irreversible inhibition, cannot be again back to the functioning. Okay, we can regain the functioning of that particular enzyme. When other enzyme molecule is synthesized, then only we can overcome the process and again the enzyme can function normally. Okay, so the irreversible inhibitors bind covalently with or destroy a functional group on an enzyme that is essential for enzyme activity, or form a particular stable non-covalent association. That means, you know, covalent bond is very strong bond. We cannot break covalent bond either breaking the whole molecule, isn't it? So irreversible inhibitors binds with the enzyme by three ways: either with the covalent bond, covalently binds with the enzyme functional group, or destroy the functional group by any mechanism, or even if it binds with the non-covalent association, this association remains very stable. Okay, so covalently with functional group, destroy the functional group, or binds with very stable non-covalent association with the functional group of the enzyme. That's why it irreversibly inhibit or halt the enzyme activity until the new enzyme is formed, okay, or supplied from the outside. Now, irreversible inhibitors are another useful tool for studying reaction mechanism. In the use portion, what are the importance of studying enzyme inhibition? We have discussed that we can delineate the metabolic steps of a process. This irreversible inhibitors also useful for studying reaction mechanism. Okay, amino acids with key catalytic function in the active site can sometimes be identified by determining which residue is covalently linked to an inhibitor after the enzyme is inactivated. Okay, so in every catalytic sites there are some key amino acid. You have already studied that, isn't it? So there are, suppose 136 threonine, in position 136 of the peptide chain, threonine is the key enzyme. Serines, various enzyme has got serine in the as the key amino acid in the catalytic site, isn't it? So if we add the irreversible inhibitor to a system, then it binds with the key functional group. By this mechanism, we can determine which enzyme or which amino acid is the key amino acid, what is the position of that particular amino acid in the functional group, which is very much essential for the action of the enzyme. So irreversible inhibitors can be used to study the reaction mechanism, also for studying the or determine the key amino acids which is responsible for the enzyme catalytic function. Okay, one special class of irreversible inhibitor is very important for your study purpose, your course, is called suicide inhibitors. Suicide inhibitors or suicide inactivators. This is also called mechanism based inhibition, suicide inhibition or mechanism based inhibition. Why? We will discuss. Suicide means one, destroy him or herself by his own, isn't it? Okay, so yourself is responsible for causing harm or to destroy yourself. That's it. That is the suicide. Here also what happened, these type of compounds relatively unreactive until they bind to the active site of the specific enzyme. Okay, so first this inhibitor binds with the specific sites of the enzyme as substrate, one of the substrate of the reaction. Then after binding only they become activated, and suicide inactivator undergoes first few chemical step of the normal enzymatic reaction, but instead of being transformed into normal product, the inactivator is converted to a very reactive compound that combines irreversibly with the enzyme. That means these type of inhibitors remain relatively unreactive first, then it binds with the enzyme as one of its substrate, then it also catalyzes the first few steps until it becomes activated and form such compound, such modified compound by utilizing the reaction's own machinery. Own machinery is used by the inhibitors. Reaction's own normal machinery is used by the inhibitors. Inhibitors binds with the enzyme at substrate, becomes activated, then follow first few steps of normal enzymatic reaction and convert the substrate to such a product that irreversibly binds with the enzyme or destroy the enzyme irreversibly. Okay, so that is why it is called suicide inhibitor. That means the inhibitor itself is a substrate, acts like a substrate, and the enzyme takes the inhibitor as substrate and follow the reaction. So enzyme itself, it produce such modified products so that this product destroy the enzyme. Okay, so enzyme itself is causing the harm to itself. Okay, so now some example. The examples of suicide inhibition inhibitors are very interesting. Okay, these are the potent pharmaceutical agent. We have already discussed because it has got few side effects, very low side effects, and that is why these are the very important pharmaceutical agent. One such agent is DFMO, difluoromethyl ornithine, DFMO. Okay, this DFMO is used in the treatment of trypanosomiasis or sleeping sickness caused by the parasite trypanosoma. For multiplication of the parasitic cell, one thing is very important, that is conversion of ornithine to putrescine, that means polyamine. These are the polyamine synthesis is very important for multiplication of the parasite like trypanosoma. And for this formation of polyamine, conversion of polyamine, one enzyme is very important, that is ornithine decarboxylase. Okay, now what happened, this DFMO, difluoromethyl ornithine, initially remain inert. So the enzyme ornithine decarboxylase binds with the DFMO. Instead of binding with the normal ornithine, it binds with the difluoromethyl ornithine, takes the difluoromethyl ornithine as substrate. Then it produces some, after binding with the enzyme, the DFMO become activated and it irreversibly binds with the help of covalent bond with the coenzyme of the enzyme system, that is pyridoxal phosphate. Pyridoxal phosphate is the coenzyme for this reaction, that is conversion of ornithine to putrescine. So when we apply DFMO, then the enzyme ODC take the DFMO as substrate, and then DFMO binds with the covalent bonds, form the covalent bond with the coenzyme of the enzyme system, that is pyridoxal phosphate, and also it binds with the some amino acid residues of the enzyme and thus inhibit the enzyme. Okay, then what happened in mammalian cell? It also can block the synthesis of the polyamine synthesis in mammalian cell too. Then why it preferentially select or effective only against the parasite trypanosoma and do not cause any harm to the mammalian cell? Because we have already said that in irreversible inhibition, the enzyme remain inhibited until the new enzyme molecule is formed. Here also in mammalian cell, the turnover rate of ODC is very high. That means the cycle, formation, synthesis, destruction, this turnover rate. So synthesis rate is very high in mammalian cells. So the inhibition is very transient to cause any harm to the mammalian cells. So it remains unharmful to the DFMO or just transiently affect the action of ODC in the mammalian cell. That is why it preferentially block the synthesis of polyamine ornithine to putrescine in parasitic cell and destroy the parasites. Okay. Now the next one is allopurinol, which is very common, and I don't know why you people always give example whenever we ask for example of suicide inhibition. You give the example of allopurinol. And so it's your favorite also, and also it's examiner's very favorite one. So allopurinol is a drug that is used in the treatment of gout. Okay, now what is gout? Gout is the excess production of uric acid, so that urate crystal is deposited in the large synovial joints and causing pain and immobility. Okay, so how the uric acid is produced? Uric acid produced after catabolism of purine. It is the final product of purine catabolism. Now Normally xanthine oxidize to produce xanthine, but when allopurinol is added as drug, then xanthine oxidase takes allopurinol as the substrate. Allopurinol at first place it is not reactive and completely harmless, but when xanthine oxidase oxidized allopurinol to alloxanthine, this alloxanthine is a potent inhibitor of xanthine oxidase. So that is why it is suicide inhibition, and that is why it is also mechanism based inhibition, because xanthine oxidase machinery itself causing conversion of allopurinol to alloxanthine, which is a potent inhibitor of xanthine oxidase, and thus inhibit the production of uric acid, and that is why it is used as pharmaceutical agent in the treatment of gout. The next example of irreversible inhibitor is aspirin. Aspirin, you are quite familiar with the name. It is very commonly used. Aspirin used in the treatment of inflammation. Any inflammatory disorders causing inflammation means swelling, redness, pain, increased temperature. This is the features of inflammation. Rubor, collar, dollar tumor, that means redness, then swelling, redness, pain, etc. That is the features of inflammation, and aspirin quite effectively relieve the symptom of inflammation. Why? Because inflammation is produced mainly by the synthesis of some prostaglandins, and prostaglandins synthesized from, you know that, from arachidonic acid with the help of enzyme cyclooxygenase. Now aspirin acetylates a serine residue in the catalytic active center of the enzyme cyclooxygenase, and thus irreversibly inhibit the enzyme. That means the enzyme remain inhibited until the new enzyme is formed. And the turnover rate of cyclooxygenase is not that rapid. That is why if cyclooxygenase is repeated by the acetylation of the serine residue in the active center by the aspirin, then the synthesis of prostaglandin from the arachidonic acid is temporarily halted and the symptoms of inflammation is subsided. Okay. Now, acetylcholine esterase. Acetylcholine esterase. Esterase means acetylcholine is formed by the ester linkage between the acetyl and the choline group, and acetylcholine esterase, it cleaves the acetylcholine to form acetylcholine to forms the choline and the acetyl group or acetate. Okay, so the acetylcholine esterase enzyme, it cleaves the acetylcholine to form the acetate in one hand and choline in the other hand. Okay, and terminate the action of acetylcholine. Now certain chemical which names diisopropyl fluorophosphate, DFP, diisopropyl fluorophosphate, binds to the active site serine of the acetylcholine esterase. I have already told you serine is very common amino acid found in the active center of the enzyme. Like also aspirin inhibit the serine residue, binds with the serine residue in the cyclooxygenase. Here also in the active center of acetylcholine esterase enzyme there is also a serine group, and the diisopropyl fluorophosphate, it binds with the active site of the acetylcholine esterase enzyme with the help of binding with the serine residue. Okay. Now as a result, what happened? Acetylcholine ester is inhibited. That means acetylcholine cannot be break down to acetate and choline. So acetylcholine as a whole is accumulated. So increase the amount of acetylcholine, and that is why overstimulation of the autonomous nervous system occurs. Acetylcholine is the mediator of stimulation for the autonomic nervous system. What is the function of autonomic nervous system? Normal function of autonomic nervous system is secretion. That means any type of secretion, salivations, sweating, watering from the eye, vomiting, etc. are the function of autonomic nervous system. It is not at all bad. In normal state it is necessary, because salivation is necessary for proper digestion, sweating is necessary to maintain the body heat, our proper watering of the eye is very important for prevent the dryness of the cornea. So it has got normal function, but due to the accumulation of the acetylcholine by inhibition of the acetylcholine esterase enzyme with the by the inhibitor DFP, overstimulation of the autonomic nervous system occurs. That means excessive secretion in the form of excessive sweating, excessive salivation, vomiting, excessive watering from the eye. In the worst case, even death can occur because of the dehydration. Because if you lose so many of body fluid in several ways, by vomiting, by sweating, by salivation, then the person can die due to the dehydration, due to overstimulation of the autonomic nervous system by accumulation of the acetylcholine. So DFP forms an irreversible covalent bond with the acetylcholine esterase. This is the basis of irreversible inhibition. It will form the covalent bonds with the enzyme or its active group or with the coenzyme. Okay, so here DFP form an irreversible covalent bond with the acetylcholine esterase, and the enzyme is inhibited until the new enzyme is synthesized. Okay, so example of suicide inhibition, please remember, example of suicide inhibition is allopurinol, ODC. These are also example of irreversible inhibition. And the example of irreversible inhibition, which is not suicide inhibition, is aspirin and DFP. Okay, please remember that. Don't give example of aspirin or DFP in case of suicide inhibition. Okay. Then the last one is feedback inhibition. Feedback inhibition is very important in the form of regulation of any metabolic pathway. Here what happened, the final product or end product inhibit the first step or the preliminary steps of any reaction, any pathway. Okay, so it is very important here because the product regulates its own production. In our physiological system, if one product is in sufficient amount and it inhibits its own production, that means there is no wastage of energy, wastage of substrate, isn't it? Because that means, yes, this product is in sufficient quantity in our system, we need not to produce this product anymore. So the product itself acts as signal for inhibiting its own synthesis, or inhibits the synthesis of its own production. Whenever it is accumulated in excess amount, it inhibits its own production by inhibiting the preliminary few steps of its synthesis process or pathway. Suppose A is converted to B with the help of enzyme E1, B to C with the help of E2, and then finally C to D with the help of enzyme E3. So the final product is D here, and if this D inhibit the E1, classically E1 or E2, the preliminary steps of this reaction, then it is called the feedback inhibition. There are plenty of example in our physiological system where the product regulates its synthesis by inhibiting the preliminary enzymes in its own synthetic pathway. Few examples are AMP inhibits the first step of purine synthesis. Purines are, you know, that adenine and guanine, and AMP means adenosine monophosphate. So here in AMP, product of purine synthesis inhibits the purine synthesis. Just like that, CTP, that means cytidine triphosphate, inhibits the aspartate transcarbamylase enzyme. Aspartate transcarbamylase is one of the first enzymes in the pyrimidine synthesis. Okay, so here CTP inhibits the aspartate transcarbamylase. So this is the example of feedback inhibition. So that's all. So in this topic, small topic, but all of them are very important. What is enzyme inhibition? What are the uses? What are the types? Very important. Reversible and irreversible. Under reversible, competitive, uncompetitive, and mixed type of inhibition. And under mixed, special one is non-competitive. You have to remember the example and have to practice all those curve, that is Michaelis Menten and lining for bark float, all of these 3 types of enzyme inhibition. Okay, we have to remember the effect on Km and Vmax. Okay, then the irreversible one, suicide inhibition. Please remember the basic mechanism and example. Example is very important. Then the feedback inhibition. So that's all. So please stay home, stay safe. Hope you are all doing well. Don't neglect your study because this course is very serious one, and you have to be sincere. You have to study. I know it's very difficult, but you have to study. Okay. Okay, that's all. Thank you. Bye.